So, very good morning. I think I'm not going to say the attendance is poor, but whoever is here, they are really interested in science, especially in the field of ophthalmology. I can take it that positive attitude, and I'm delivering the talk. Good morning. I'm Dr. Sundaresan. I'm heading the Department of Genetics. For the past 25 years, I'm working in the field of ophthalmic genetics. So you name any kind of disease from cornea to the optic nerve, we extensively studied, and then we published more than 160 papers, and I produced more than 25 PhDs, and really you are interested, please go through our publications. So lack of time, what I'm going to do what I did for the past 25 years in the field of glaucoma. So that's what I put the title, A Journey in Glaucoma Genetic Research. I think concerned with the glaucoma, the ciliary body secreting the aqueous humor, and then from the posterior chamber to anterior chamber, and then it is draining out to the abiscular vein through the TM and SEMS canal. And then there are so many other, the glaucoma is an irreversible blindness due to the dysfunction and the damage of the retinal ganglion cells. So once the extracellular matrix blocking the meshwork, it increases the intraocular pressure, and then the patients lose the peripheral vision. In addition, the restriction fluid movement in POAG or in the steroid-induced glaucoma, it is the reason because formation of the cross-linked actin network, abnormal development of extracellular matrix, and then decrease in phagocytosis. In addition, central corneal thickness, there are so many other reasons for formation of the glaucoma. So we extensively studied primary open angle glaucoma, primary angle closure glaucoma, pseudo-exfoliation, and primary open angle glaucoma. So concern with the myocilin gene, that is the first gene, is the candidate gene for primary open angle glaucoma, that is identified by the Dr. Stone group from University of Iowa in 1998. That time, the entire world thought about is the only one gene. Now the scenario is different. It seems to be a very, very complex disease. Glaucoma is a very, very complex disease. Now they are telling instead of complex, it is an incredibly complex disease. That's what I am putting a pathway here. There are so many pathways are involved, and then it is very difficult to understand what exactly the pathogenesis. So you can see the yellow dots here, highlighted here. There are so many important factors associated with the primary open angle glaucoma. We are working on one particular dot to understand the pathogenesis of primary open angle glaucoma. So what we did for the past 25 years, we recruited huge number of primary open angle glaucoma patients, and then more than 500, and then we screened a specific gene that is called myocilin. You can see the exon 1, 2, 3. The exon 3 is the hot spot. There are more number of pathogenic variation we identified in the exon 3. And then what we did, when you observe a particular family, this is an autosomal dominant disease. Based on the volume of the file of the patients, we can able to say the severity of the disease. So one particular, lack of time, I'm going to focus on only one particular mutation in the myocilin, that is nothing but the glycine 367 arginine, meaning you and me, if the intraocular pressure is normal, that is we have glycine in the position of 367. So because if there is any mutation, it is instead of glycine, it will convert to arginine. So based on the change of the protein, what it will do, it will accumulate or it will interact with other normal and abnormal protein. It will form the extracellular matrix. That matrix is blocking the SEMS canal or the TM. So that's what we are getting the increase the intraocular pressure. So what we did, we have taken a vector that is called pcDNA3, that is a kind of vector, and then we cloned the entire myocilin gene, and then using the side-directed mutagenesis, we changed the glycine 367 to arginine. And then you can see the panel 1, 2, 3, untransfected, and then wild type, and then glycine 3, the mutant. Meaning, so the untransfected in the sense, we didn't put any DNA, but in the wild type in the sense, the wild type myocilin is transfected in the TM cell. This is the TM cell, what in our lab we have International Eye Bank. So we'll take the TM and then we'll put into the culture. We can make a primary culture derived from the TM. You take the TM and then you can do the experiment. That's what we are doing. So in the wild type, the myocilin, and then another you have to understand that Gerald talked about the gene and gene protein. So the expression of the protein is very, very important. Some of the protein, it will come out of the cell. Some protein will sit inside the cell, but the concern with the myocilin, it is a very leaky protein. Some proteins sit inside the cell, some proteins come out of the cell. If you measure the TM, then you can isolate you can detect the protein outside the cell or inside the cell. So you can see here the comparison between the wild type and then the mutant. You can see the green signal or exclusively the myocilin. The signal is more in the mutant than the wild type, meaning we proved using the Western blot with a specific antibody. So you can see in the wild type, the intensity of the band is thick, both in the intracellular and extracellular, as I told you before. But in the mutant, you can see in the intracellular, the protein, the intensity is very high than the extracellular, meaning all the protein is, so once it is mutated, it is sitting inside the cell, it is not coming out of the cell. That's what you can see the intensity of the green signal in the wild type is very, very high. So due to this, the extracellular matrix is blocking the mesh work, increase in intraocular pressure, peripheral vision loss. Okay, so now, I think you know about the liquid biopsy. The same way we have proved the control and cataract. During the surgery, just take the aqueous humor and then measure the protein. For your kind information, when you measure the aqueous humor, especially in the primary open glaucoma, the TG beta 2 level is very high. But in addition to that, here the primary open glaucoma, you see the intensity between the cataract. The cataract and POEG, the intensity is very high, meaning the amount of myocilin is very high in the primary open angle glaucoma patients. And then this is the long story. Once the gene is identified, the entire world is, very happy. It's only one gene. That time, we made a model. Nobody made the model for a computer model. So what we did with the collaboration with the Madurai Kamaraj University, Dr. Krishnaswamy, we made a face building model for the entire myocilin. That's what we did it. And then I told you before the TJ beta. So we are collaborating with all over the world, all the glaucoma groups, and we did the genome association studies or the target sequencing and everything. So here you can see I told you the content of the TJ beta is very high in the aqueous humor. Here genetically we proved that TJ beta receptor 3 is very significantly associated with the primary open glaucoma. I can give you another information about TJ beta. You can see, if you see the TJ beta 1, 2, 3, and then TJ beta receptor 1, 2, 3. Here, according to the genetic screening, we got more association in the TJ beta receptor, and then we published this paper in 2015 in Human Molecular Genetics. All the authors, I think they are the leading glaucoma groups all over the world. And then, interestingly, there is another gene that is called SIX6. This is another candidate gene. Now, for your kind information, myocilin is not only the one candidate gene. There are 4 candidate genes already identified. In addition to the candidate genes, more than 540 associated genes are there. It means that's what we are telling, it is incredibly complex. So this particular gene, we did the genome association studies. You can see the round. We got the hit in the SIX6 is one particular gene, is closely associated with the primary open glaucoma. Not only primary open glaucoma, it is also associated with the normal tension glaucoma. Then when we screened our cohort, when we screened our cohort, we identified lot of variations. Interestingly, in the promoter region, there are 4 base pair deletion is there. So that is a very, very interesting story. You can see this is the promoter, the enhancer sequence. I can put it here. You can see the ATCT. You can see it in the center. So that is nothing but the 4 base pair deletion is deleted in the particular patient. Now, if you look carefully in that particular region, there are more than 4 transcription binding domain is there. It means once the particular 4 base is deleted, lot of, it won't synthesize the right protein. So that's what, the severity of the disease is very high to this particular patient. So then what we did, again, we cloned the gene, we deleted the 4 base pair, and then we have to prove it in the GFRA-fish model. That is the biological relevance. What exactly you are doing in the bench, you have to prove it in the animal model. So here we have taken the GFRA-fish model. So this is the one. One is the wild type, and then the mutant. We created 2. You can see, especially in the GFRA-fish, please observe the globe. The globe size is normal in the control, but in the mutant, the globe size is reduced. You can see in the panel F1, especially E and G. So the globe size is big in the normal, and then in the mutant the globe size is reduced. You can see the retina. So especially the ganglion cell layer, amacrinal cell layer is very normal in the control, but here in the deletion one, it is completely, shrinked, or it is reduced the retinal thickness. So through that, we proved both genetically and then with the biological relevance with the GFRA-fish model. We published this paper in Molecular Genetics and Genomic Medicine. These are our collaborators. And then, interestingly, we supposed to make a DNA chip for diagnosis of glaucoma in India. So that's what, we made a collaboration all over India, the leading institution, and more than, we collected not only in our place, we collected north, south, east, we collaborated with all the leading institution, and then we collected more than 1,000 sample, and then we screened what is the exact the variation within the population. But because of the heterogeneity, the problem is the heterogeneity. Because of the heterogeneity, we can't be able to find out the common marker all over India. So now we are adopting another strategy, like you can recruit the patient, and then based on the intraocular pressure, meaning when the age is less than 35, if it is a juvenile open angle glaucoma, intraocular pressure is very high, definitely they are positive for myocillin. So like that, you can able to screen, and then we can go for counseling. That is the story. Gerald, I will take another 2 or 3 minutes, please. This is another family. It's a very, very interesting family. More than 5 generations, and then more than 400 study subjects. Out of the 450, 20 of them are primary open angle glaucoma, 20 of them are suspect. So we did the entire genome-wide association studies, or we screened the whole exome sequencing, and then we can't be able to identify any mutation in the known genes or the associated genes. That's what I told you before. 4 candidate genes for glaucoma, 450 genes are associated genes. We can't be able to identify any mutation. It means there may be an additional candidate gene for this particular family. Then again, we screened through whole exome sequencing, both intronic and exonic region, we entirely screened. Now, the what we identified, that is called rare variants. The rare variant is associated with this particular family. That is the low frequency of the pathogenic variants in multiple genes is playing a role for this particular family. That is the ultimate outcome of this particular study. Now, this is, as I told you, this is a JOAG family. You can see is autosomal dominant disease. Is a vertical transmission, or any one member is affected in this particular generation. Obviously, this is the myocillin mutation because the intraocular pressure is very high, the onset is less than 35. Now, this is the current recommendation for JOAG and POAG. If it is a JOAG, onset before age 35 with the family history, with and without family history, we can go for myocillin. Or it is a primary open angle glaucoma, onset before 50 years, with the family history, we can go for myocillin. This is the recommendation worldwide to screen the myocillin. I think in addition to that, we extensively worked on angle closure glaucoma. We identified the candidate genes because angle closure glaucoma, there are 2 schools of thoughts. One is like anatomical problem. It is not a genetic problem, but the scenario is changing now. 60% of them is anatomical, and 40% of them is genetics. So we recruited all the cases, and then again, it's a collaborative project. We collected 10,503 cases, and then 29,567 controls all over the world, in 20 countries, and then we identified the candidate gene for angle closure glaucoma. Not only candidate gene, the loci identified for angle closure glaucoma. Okay, this is the one. In addition, as I told you, if you are a clinician, please, whatever either the aqueous humor or vitreous humor tear is very, very important. All the biological fluid. Through that, you can find out. So what cytokine is, correlated with the pathogenesis. That's what we did here, and then all the informations are here. Due to lack of time, you can read our publication in the current eye research. So I think very recently, I think we got a major grant from Indian Council of Medical Research. So my collaborator is Aparna Rao. So here we are making a kind of different strategy in the sense. So you have to take exactly where the pathogenesis is going on. You have to deal with that particular disease tissue, then only you can able to understand the pathogenesis of any kind of disease. So here our proposal is, what we proposed, suppose when you do the tuberculotomy, so I think is a kind of invasive procedure. You are not taking only the TM, you are taking along with the sclera. So when you grow it in the lab, the sclera cells is growing faster than the TM. So you can't be able to deal with, because our aim is to make a single cell RNA sequencing to find out what exactly the pathogenesis. So here, Dr. Aparna is very good in this particular surgery. She made a review article, and then she published that one. Anybody is interested, go through that. So ultimately, maybe I can present this particular data in the next meeting. So I think I have collaboration concern with the glaucoma all over the world, and including in our hospital. I'm collaborating all our glaucoma specialists with my students, and I think with this I will stop here. And finally, this is my slide. Even I put a slide, again I'm correlating with the science. You can see in the center, you can see the light, and then the periphery is, not that much. It means the glaucoma patients, they are losing the peripheral vision, they are getting only the central vision. So our aim is to, at least, why not you bring back the vision to that kind of patients through our research. Thank you very much for your attention. Thank you.